Hi everyone, welcome back. And in the last couple of videos, we established a little bit of groundwork for this video where we talked about, initially we started by talking about voronoi. We understand what it is now, hopefully. If not, go back to video number 273. And in the last one, we did a step-by-step approach of understanding or establishing the concept of object segmentation using this new voronoi-based segmentation approach. And it's pretty straightforward. All we are trying to do is find where the objects are in a way by finding the maxima within each object and then applying voronoi to divide these into different regions, to divide our image into different regions. And within each region, we're trying to segment the objects, and that's how we get object segmentation. And all of that, now that we understand exactly how that works, now let's convert everything into a single line approach. That's it. One line, thanks to pyclesperanto library. And again, no machine learning, nothing needed, because this is not a super duper complicated example. You're not trying to segment glomeruli in pathology images or trying to segment some satellite images for trees and roads and so on. This is pretty straightforward application where you have a background and you have a bunch of objects in the foreground, and what is the best method to segment them. And after segmentation, let's also go ahead and look at some analysis using our plane, pandas, and seaborn visualization. So that's the plan for this video, and I will not try to make this very long. So let's go ahead and jump in. And I already mentioned this in my previous video. I am going to use pyclesperanto library, and to find this, you can just go look at the link down in the description or in the file that I'm going to share with you. Or if you just want to do it yourself, go ahead and Google search for this term, pyclesperanto prototype or Python pyclesperanto. It'll take you to this GitHub page that has all the information about how, and also they've done an amazing example in providing a lot of examples right here. They have done a very good job in providing a lot of examples here, and I am just stealing a couple of these examples to let you guys, to train you in a very visual way. That's all I'm trying to do. Okay, now let me jump back to my code. And for this exercise, I am going to use a scientific image, a image that comes in the form of CZI. This is from a Zeiss microscope, but if you have other type of microscopes or other images, obviously the process applies. But I am using this deliberately to show you a couple extra steps, right? You learn many things in my videos, so I want to include these. So what is this image? This image is, as you can see, a whole bunch of cells, cytoplasm in green, nuclei in blue, and the orange stuff is, I believe, mitochondria. So if I turn one thing off, these are all individual channels. So this image is, believe it or not, this specific image is grayscale image. I'm applying a green color so that it is visible to me. Yeah. And this image has three channels. One is the one that shows the cytoplasm, the other one that shows the mitochondria, and the other one that shows nuclei right there. My goal is to analyze these nuclei, which means my interest is in channel number 3. Okay, this is how you deal with multi-channel images. If you have a multi-channel satellite image, because you may be imaging it under multiple bands, select the band of interest that you are interested in, and then go ahead and perform these analysis, right? So anytime you are dealing with scientific images, the images are not just JPEG or PNG because they need more dimensions to capture additional information. There you go, a little bit of scientific information. If you're non-microscopist, hopefully you'll appreciate that. Okay, so for the CZI file, I am going to use the Zeiss's CZI file library. Pretty straightforward, pip install CZI file. And let's go ahead and start by understanding the version of Python. I'm working on 3.7, just in case you're curious. And also, I'm going to import my CZI file library in case you wonder what version it is. It's 2019.7.2. That's the version. Okay, the other ones are pretty straightforward. Matplotlib, Numpy and pyclesperanto prototype. We used this in the last video anyway, so you should be familiar with this. And this is exactly what I was showing you right here, this GitHub page, and follow the link to install this. It's pretty straightforward. You just kind of install your PyOpenCL, and after that, go ahead and pip install your pyclesperanto. Worked very well for me. Jumping into the code, let us import this library. There you go. And let's also import scikit-image, a couple of methods from scikit-image. Okay, now let's start by importing our image. Again, my image is a .czi. You saw that it's a 3-channel image. So let us go ahead and imread. And if you look up here, it shows a bunch of additional stuff, right? So it is, I believe, the first one stands for the time series, like, because this image can be a time series. As the time goes by, how are the cells or nuclei moving, or how are the cells evolving, and so on. So that is the time series, the first one. The second one is, I believe, the scenes, which is, this is scene number 1. I have another scene where I have a different experiment going on. So that's the placeholder for that one. And this 3 represents our 3 channels that we have. In the third channel, I have nuclei. This is my image X and Y dimensions. And the last one is the Z. I don't have like a Z or Z if you're from Canada or non-US locations. So this is just an X and Y image. So that's the explanation for this image. All I care about is nothing right there, nothing right there, all pixels right here, but in the third channel from here. So let's go ahead and extract that. Yeah. So if you look at the image shape, that's exactly what it's going to print down here. Yeah. So let us extract the third channel. So right here. So sorry, let us extract only the ones that we care about, which is the number of channels and X and Y. And after doing that, I am only going to extract the third channel. I already showed you this in a different software, but let's look at these images. Channel number 0 is showing me it's exactly this image. It's exactly this image, right? It's showing me the mitochondria. And let me minimize this. And channel number 1 is my cytoplasm right there. And channel number 2 is the nuclei that I care about. So let us only keep that and remove the other 2. So I'm calling that DAPI. Yeah, let me not get into the details of why it is DAPI. Go Google search for DAPI, you'll know a lot more about it. So I am only taking that, and let us look at the data type of that. So this image is stored as unsigned integer 16. We do not need all the 2 to the power 16 depth, 65,000, whatever, 535, 536. We do not need all that depth, so we can convert that into 8-bit image. So I am going to convert my image into 8-bit. And I am also rescaling the intensity right there after conversion. Otherwise, your intensity would be like some weird values, and it doesn't work out that well. So again, all of these I covered in the basics of Python, basics of image analysis on my in my channel. I don't want to make every video about image analysis lectures. So let's go ahead and look at this image. This is exactly where we are right now. If you already have an image that looks like this, a single channel, ignore whatever I just showed you right now. Just start with this. Go ahead and imread your image and start from here. Yeah, this is just extra stuff that I'm giving you. Okay. So now comes implementing this. So in the last video, you saw that we can use GPU for Clesperanto computations. So let's first go ahead and look at what available GPU devices we have on this specific system. The system that I'm working on is Quadro P5000. I have 2 of them. So let's go ahead and assign a device. So my device is clesperanto.select device RTX. So let's do that, and then print the used GPU is that one. Okay. Again, if you do not have GPU, if you're not working with these multi-channel type images, if you just have single images, everything is useless, whatever I just showed you. Start with imread and then directly jump on to this part. That's it. In fact, the actual meat of the entire code is just one line, like I promised in the last video. I'll get to that in a second. So let's move through this pretty fast. So we did the GPU, and now I am going to push my image into GPU, so it's accessible via GPU. So cle.push, let's go ahead and do that. And let's print the image in GPU size. It should be exactly the same as before, right, in the CPU. So okay, now let's go ahead and show it, and then it should look exactly the same. So this is the one that I pushed to my right there GPU, and there it is. It should be exactly the same between CPU GPU. Okay, now comes the actual part, right? You have your image, and now you want to segment it. Anticlimax right there, just single line right here. That's it. So within pyclesperanto library, I am going to use voronoi otsu labeling onto this image. It automatically does a bunch of stuff. What does it do? It finds where these peak maxima locations are for each object, and then it divides the image into different voronoi regions, and then it applies a mask within that voronoi region to segment your images using otsu, and then it labels them. So what the output image would be is basically a labeled image. It does a lot right there. Let me create some room so we can understand that. And there are 2 sigmas that we need to provide as part of your voronoi otsu labeling. And if you remember from my last video, I asked you to think of 2 different sigmas that we have. One, we need to blur our objects so we can find the peak locations. The other, we need to blur the objects so we can properly perform otsu thresholding. So these 2 sigmas control exactly that. So go ahead and play with this if the default values do not work for you. I always play with this to get a good balance in terms of my segmentation. Okay, so that exactly is what you probably tuned to this video. Oh my God, if I can select this. Okay, there you go. And voila, there you go. That's an amazing segmentation right there. Yeah, we did not do much. All we did was a single line right there. Now you can remove the ones that actually touch edges, which is customary if you're doing object size analysis. So for that, in pyclesperanto library, you have exclude labels on edges. So you can click on that and then go ahead and show this. So you can see some of the edge ones are gone, and now you have the ones that are not touching the edge. Now you want to know exactly how many of these you have. So within pyclesperanto, maximum of all pixels, how many do you have? Let's go ahead and print them. I have 110 objects. Okay, now you can go ahead and save this image. Again, this is another thing that you may not easily find by looking at the documentation on pyclesperanto. You may have to dig a bit deeper. But if you want to save this output image into your local drive, then it is first thing you need to pull this from wherever it's working on, right? Pull this out of this pyclesperanto. So it's remember we pushed it, now we need to pull it back. So cle.pull segmented excluding edges. This is the image that we just created, and then go ahead and save. That's it. Let's go ahead and show the image. It should be exactly this. I mean, it's exactly the same image right there, except this is labeled now. I hope you know what labeled is. Object number 1 is assigned a value of 1, object number 2 value of 2. So as the object number goes, then we have 110 objects. So the 110th object will have a value of 110, and that's why it is much brighter right there. And these images, I believe, are do I have this here? Yeah, unsigned integer 32. Why? Because you may have a lot of objects in an image. If you only save them as 8-bit, then your maximum number is going to be 255 for your object. It's storing them in unsigned integer 32, meaning you can have 2 to the power 32, that many objects in a given field. Good luck if you have that many objects in a field anyway. So let's you can go ahead and save this. Now, there are a lot of other plotting options as part of pyclesperanto. So let me just go ahead and show you a couple of these pretty cool useful functionality, I should say. So first of all, pixel count map, meaning you want to plot each object based on its size. In this case, they all look very uniform, but typically if there is a range, this can be a pretty good visualization. So there you go. All the red ones are larger, all the ones in green are small, in the blue are smaller. So that is one way of plotting. Extension ratio map is another one. So this is basically, it's derived from the maximum distance of pixels to their object's centroid divided by the average distance of pixels to the centroid. So another morphological way of actually showing these variations right there. Mean intensity map, again, in this case, it's probably all of those have pretty much the same intensity. No, there seems to be some variation. This is much brighter apparently in the original image. So if I go back to the original image, wherever that original image is, yeah, you see these ones are brighter than the other ones. So it's basically the pixel brightness right there. Minimum intensity, same as max. So I can go through all of these: max intensity, standard deviation intensity, where the intensity is varying within each object. This can be another good one. So apparently right in there you have a lot of variation. I'm curious now. Let's I don't want to extend this way too much, but yeah, in that object apparently things are changing a lot. I see that even here. You see, so this can be sometimes very useful in identifying objects where things are going on within that specific nuclei. If something is going on, then if it appears brighter, if it appears that there are certain things going on, then this map can be a useful way of actually finding that out. Neighbor count, I guess, based on the number of neighbors. So it is showing us the voronoi right there. Touching neighbor count, proximal neighbor, so you can play with all of that. Okay, let's use what we have and extract statistics. How do you get statistics out of this? Within pyclesperanto, go ahead and do statistics of labeled pixels, and it dumps a whole bunch of statistics that we can use. So if I go back and look at my statistics right there, you can see it's a dictionary. It has like a bounding box dimensions in case you need bounding boxes for each object. It has minimum, distance, mean, max, pretty much anything that you can think of from a statistics point of view, and you know how to deal with that. I usually convert that into some sort of a pandas data frame. Go ahead and describe it. Go ahead and look at what columns it's reporting. A lot of information right there. And just plot a couple of them. Let's look at the area. So what does the area look like? What type of distribution? So there you go. That's the distribution of area. And let's look at the mean intensity. Again, you know how to do the plotting, but I just want to show you a couple of these. And if you want to draw bounding boxes, what was I trying to do here? Yeah, convert this object to Numpy array. So yeah, that's another important thing. So if you have your image right there, and if you want to get your bounding box values, so I picked bounding box minimum X right there. So I'm just extracting those right here. So if you go minimum X right there, there you go. This is all the bounding box X minimum. Similarly, extract all the other values and then go ahead and plot it on the image if that's exactly what you want. Shall I show everything? Why not? Let's run pretty much everything here. I was trying to wrap this up video, but there's a lot of interesting stuff that I really want to talk about. Okay, so I got my bounding box minimum X, minimum Y, maximum X, maximum Y, and we are going to take these and iterate through each of our objects, right there, and then plot them onto our original image. So that's exactly what this is doing. I'm using OpenCV for that. And there is the box. You can draw the boxes in red, green, blue, whatever color you want. And I can right away see that, okay, maybe, oh no, that is 2 nuclei. That's doing a good job. Maybe some of these nuclei are divided into 2, like this large one is divided into 2. So this is where I can experiment with my sigmas to see how much of over segmentation versus how much of not doing a good segmentation. I'll leave that exercise for you, but I hope you found this to be useful. And again, before jumping into deep learning, if you want to get a task done, see if there is any classical ways of getting it done. And there's probably an amazing library out there like pyclesperanto for your specific task. So first investigate that and then jump into deep learning. I'm a huge advocate of deep learning, but I think if an application doesn't require that, then Occam's razor, guys. Whatever the easiest path, easiest solution is, that's exactly what you should be following. Thank you very much. In the next video, let's extend our knowledge of this, and I'll show you how you can get segmentation done in less than 10 lines of code, and hopefully I'll live up to the expectations. So please stay tuned for that one.